Hello, ma'am. Good morning.
Good morning, Daniel.
How are you doing?
I'm good.
Yeah. Great, great. So I see so many participants today, and also we have few participants on YouTube stream as well. Since we couldn't accommodate everyone in the Google meeting, we had really good response. We had around 200 plus registrations.
That's so nice
So
to hear.
yeah, so we couldn't accommodate everyone. So it was first come first basis. So
Yeah.
we have allotted few people in Google Meeting and few people on YouTube. So the YouTube link is also unlisted. So the participants who will get the link can only see it. Not every it's not like everyone cannot see.
Okay.
So yeah, I hope I am audible clearly.
Yes, you are.
Okay. Thank you so much. So, uh, on behalf of the SciComm Synapse, I welcome you all. And I would like to give a very short introduction to our community for the people who don't know it, and also for ma'am, since it's very new. So, we few youngsters, I would say, from different backgrounds of science have started this science communication group where we invite various professors and we try to discuss the journey of science. We also try to invite various PhD students who are already in abroad and know their like what was their trajectory, and also few postdocs or some research associates in industries as well. So we are in building a community where we do something uh, after work, I would say, something which is apart from science and science communication, which I think everyone would love to do it. And nowadays, people are also taking interest. So we are growing with the help of our participants, especially. So thanks for joining, everyone. And now I would like to tell Bhakti to kindly introduce ma'am very shortly, and we can then proceed ahead with the event.
Yes. Hello, everyone. Am I audible? Okay.
Yes.
So let me just quickly introduce our today's speaker, Professor Devarshi Gajjar, an esteemed scholar and seasoned professional in her field, holding a master's degree from the Maharaja Sayajirao University of Baroda in 1999. She completed her PhD from Sardar Patel University, Vallabh Vidyanagar in 2002. She worked for a decade as a scientist at Iladevi Cataract and IOL Research Center, Ahmedabad. Then she joined Department of Microbiology and Biotechnology Center at the Maharaja Sayajirao University of Baroda in 2013. Currently, she is working as a professor and also as a coordinator of Biotechnology Master's Program at the Maharaja Sayajirao University of Baroda. Her expertise lies in medical microbiology, clinical research, and antibiotic resistance. She has presented her research at many national and international conferences, making her research global. She also received Women's Scientist Fellowship from Department of Science and Technology, Government of India. Professor Devarshi Gajjar's research primarily focuses on understanding bacterial and fungal pathogenesis, molecular mechanisms of antimicrobial resistance, biofilm formation, and technologies to detect and combat drug resistance. She also understands the importance of science communication and is active in motivating students to pursue career in the research. She is a strong, confident, and family oriented woman and also a great parent. So, with this warm welcome, now I request Daria to continue the event.
Thank you, Bhakti, for the introduction. Ma'am, I hope the introduction was correct.
Absolute. Yeah. Perfect.
That's great. Bhakti did a lot of effort in finding out your introduction. Okay, so um, I hope that you are ready for a short question and answer round, quick with
Yes,
us,
I am.
where we will try to understand that what is the situation of women scientists currently in India, and also there are very few young professionals as well as UG PG students. So maybe you are, maybe your students would also be present there. So your
Yeah,
journey would
so
also get new for them as well.
yeah.
Okay. So the first question that I would like to ask you today is that, like, what initially sparked your interest to pursue research, and like especially when.
Yeah, so let me begin. Obviously, we are all biologists, so that means we don't like mathematics, or we don't know how to do. We are not very good at mathematics, so we chose biology. And obviously, all those who choose biology want to become a doctor on the first place, right? But then you don't get into medicine, so you choose another option of becoming a doctor, that okay, you can still become a doctor by doing a PhD. So that was it. Like, I could not become a medical doctor, so I thought, okay, let me do a BSc, then let me do an MSc. It just fall in place. Regarding research, yes, my PG exposure in the Department of Microbiology and Biotechnology, when I was doing my MSc, we have a very rigorous MSc dissertation. And in those days, it was for almost 2 years. So we used to start dissertation very in the very first or the second month of our joining the department. So I think that was a big turning point, and that was a skill that I came to know my how much I can stretch myself, how much I can work more, how good questions can I ask, how can I comprehend a particular science topic, and can I ask the relevant questions, and can I execute experiments for those relevant questions. Yeah, so I think that was my MSc was my turning point, and in my MSc I was very clear that I am definitely going to do my PhD. During BSc you are very naive and you don't know, and there are so many students. BSc has around 200 to 50 students in each class, so you are lost and you don't know what you're doing, you're young. But yeah, MSc, the group is small and you can interact more, you can share your thoughts more. And yeah, so my MSc was my turning point, I will say that. Did I answer it properly? Yeah.
Yes, absolutely. And like uh, were you sure at that point that okay, like you would become a PI, because your journey was quite trajectory, also went to the industry. So
No. Yeah. So no, I just wanted to do a job and I just wanted to survive. We are all from very much middle class backgrounds, and all you want to do is become independent. Your parents want you to become independent, do a decent job, and I shifted fields. So my MSc dissertation was on crude oil biodegradation. My PhD was on a plant virus purification and characterization. So I was into agriculture and I was into tobacco growing, tissue culture, and doing that. And my first job after PhD was in an eye hospital. And it's just my fate, and that took me there actually. And मेरी चप्पल घिस गई थी नौकरी ढूंढते ढूंढते अहमदाबाद में, to be very honest, after my PhD. And I will be brutally honest that they don't give jobs to women. Even today it's the same thing. I am the placement coordinator in my department, and every now and then we hear that okay, we need people for night shifts, and therefore women are not like the ideal choice. So, and yeah, it's very sad, but that's the fact that I gave interviews at almost all the big companies in Ahmedabad, and I could not be placed over there after my MSc, after my PhD. So, and particularly because I had an agriculture background. But thanks to Dr. Abhay Vasavada, who saw the spark in me, and he offered me a postdoctoral position, my first job. And then nothing could stop me. Then I started writing my own grants. I got my ICMR research associateship first, then I got the women's fellowship second, and then I became a recognized guide in the Manipal University. So no, and even while I was working in Ila Devi, I never thought I will leave Ahmedabad. I thought meri zindagi, matlab I will be there all my life. That was it. But then this position came up, and MS University thought I was worthy of it. So I shifted to Baroda in 2013. Yeah, so no, not even in my dreams I had thought that I will become a PI one day.
That that's a very fascinating journey. I mean, I would say that today's young professional expect that they would easily get a job after doing a PhD. But let's say if
Oh,
10 to 20 years back, if you were struggling to get a job, then the scenario of the job market right now is much more competitive and difficult.
yes, certainly. Yes,
So
yes.
I would say this is an free advice to young graduates or who are going to graduate soon. Like one of me, maybe after 4 years, I'll also finish my PhD. After 3 years, then would be a tough thing for me. Yeah,
So
and
you should never be scared of shifting fields. Like I see this young generation, they come and tell me, I only want to do neurobiology. I'm like, how can you decide you just want to do neurobiology all your life? You should explore other fields. So it is my personal experience and personal advice that one must not restrict to a particular field. You should take the risk and you should shift from, and it helps, it really helps when you shift from one field to the other. It might take time to adjust, but then it helps. Yeah, sorry there. Yeah.
absolutely, I was always working in molecular biology, always or immunology, and now I'm completely doing like cell biology and virology, which is like my PhD topic, completely different. So yeah, I get your point. And would you like to share some memorable incidents since you mentioned upon your rigorous master's thesis at MSU? Would you like to share some any like memorable incident maybe with your colleagues at that time, or maybe something for research?
I don't know whether. So what I remember is back then. Even today we give paper presentations, right? So back then we also used to give paper presentations, and sequencing was just upcoming, and that was so fascinating for all of us in 1999 that the human genome was going to be sequenced. So we were very much fascinated and excited about the whole thing, and we were convinced by our teachers. And I will say more than 90% of role of our teachers, that they convinced us that the next 20 years are going to be good, and if you just stick to the field, you will survive. I think that's the only motto one should have, that you will survive. And we were convinced by our teachers that yes, we will survive. So MSc was very, very, very good, and my topic was biodegradation, as I told you. So I had two cultures, and I still remember that day. One culture was Pseudomonas, and it was degrading benzene. The other one was some other name, and it was degrading naphthalene. And I was supposed to do protoplast fusion and make another third culture which would degrade both of these compounds. And I could actually do that, and that day was so, everyone was so happy, my mentor was so happy, and it was really very, very good. So that was one thing. And again, science convinced me that biotechnology and microbiology is the field where you can explore and you can do new things, and you feel good about doing new things because you're solving some problems. That is one incident. And the other incident, as in how I shifted to antimicrobial resistance, is in Iladevi Cataract and Eye Research Center, when a lady got Pseudomonas infection, and it was so multi-drug resistant that the eye had to be removed from that lady. So that just touched me. And I had I had many sleepless nights, and I felt that as a microbiologist I could not cure that Pseudomonas, and I could not give the proper drug to. And that is how I started doing antimicrobial resistance, and I started reading more on it. And now we know that how difficult the problem is. The whole world acknowledges that it is a very big problem. So yeah, I think these two are the two main incidences for me to stick to to science, and I can spend my entire life just reading manuscripts and yeah, enjoying them. I just like reading, and I can read reviews after reviews, line to line, word by word, and I will not get tired doing that.
I think that's again a free advice for all the researchers out there to start reading.
Yeah.
One of them is me. I don't like reading much. I directly go to result section and then see, oh, is there something interesting in figures? Maybe. If not, then okay, next paper. But that's true. And like, uh, so like your transition from masters to PhD, since you say that you went to an agriculture field. So like, how
Yeah.
did you transit during that point, and during that time, what was like a criteria? Right now everyone looks for CSIR and these kind of exams, but back then, you know, how was people interviewing for PhD?
Yeah, so even during our time, everyone wants to be in an elite institute, right? Even I had dreams of going to IISc, going to NCL, going to IITs. So we used to give interviews everywhere, and but then because there were not many ICERs and NITs, and the IITs were only four at that time. There were only the main two, four main IITs were there. So getting into these premium institutes was very, first of all, difficult, and the waiting period to register as a PhD student was very long. So I did get into NCL, and there the waiting period, because all the positions for doing PhD were filled. So until and unless somebody graduates out, you cannot register. So the waiting period was around 3 years. And then my dad put his foot down that you cannot wait for 3 years and then start your PhD. That's not a very practical decision. You must take a degree as early as possible, because the real life starts after you get your PhD degree. At that time, I might have cursed him for not allowing me to join NCL, but now I feel whatever happened for the best. So then I got into a project position in Sardar Patel University. So it was a collaborative project with Gujarat Agriculture University, now the Anand Agriculture University, and SP University. And I was a JRF, and I just registered in 2 months, and that's it. At in another 4.5 5 years I had my degree. So it does help, if I think my dad was right that if you want to do a PhD, why do you want to wait for it? You just do it. Just don't wait. I mean, don't wait for the right lab. It's just a degree. And I feel that yes, it does matter sometimes to do it at a good place, but I don't know, it's all, I mean, in what's written for you. So yeah.
Absolutely, absolutely. I will agree on this. The fate matters.
Yeah.
Great. So, like, of course, the PhD would have been a lot of struggles and to generate the data, publish the manuscript, getting funding in the lab and everything. So, like, what was the I would say what was the biggest struggle that you had during your PhD, like, or as a part of research, or for funding, or like, what was the crisis during your PhD? The major problem you faced?
So I will say that I was really blessed that my guide was Professor IL Kothari, and it was just like a family in SP University, that your lab is your family, and you never struggle in the lab. But it was a collaborative project with Anand Agriculture University. So the struggle was to, shuttle between the two institutes and manage the time. And because the ultracentrifuge for the virus purification is in Anand Agriculture University, you are doing an SDS page in your lab at Vidyanagar. So for 16 hours, the centrifuge is running there. You collect the sample, you come to the lab, and then you start. So you before that, you have already have to cast the gel, keep it ready, So all these running around things, but I enjoyed it. I must say I really enjoyed it. And fortunately the lab was full of antibodies. So the other PI, Professor Ashok Mishra, had just returned from Australia, and he had got all the antibodies against the viruses. So all we had to do is just purify the virus and check all the antibodies that were there and characterize it, that which family does it belong to, and then do an electron microscope. So we didn't have an electron microscope, so that was another struggle, to look at places who has the electron microscope, how do you carry the sample there, and how do you so it's just the technical glitches that I think I had struggled. But otherwise, with respect to the facilities, I think we had, the project was a big project. So, and it was funded by ICAR, Indian Council of Agricultural Research. So we had enough money to do the things. But uh, uh, because it was the first time somebody was purifying the virus, somebody was doing an electron microscopy, so you had to read everything. And in those days the photographs would come in the, you have to develop the photograph in a dark room, which also I have done. That you have to learn the whole process of developing the photograph in a dark room. So yeah, it was fun actually. I just like the fact that I'm learning something new that I never knew. So that was always my motto, that am I learning something new that I never knew before? And yes, that answer was yes. Even when I came to Ila Devi, there was so much to read about the eye. So again, the same question, kuch naya janne ko mil raha hai? Yes, absolutely. It was the best place there for all the ophthalmologists. So I was learning so much from the doctors. Yeah.
That's so good. At that time, was there an electron microscope in Gujarat? I don't
Yeah. So
think.
yes, the, what is it called, in Vidyanagar only, it had just been installed, the instrumentation facility. And it is still there. The TEM and the SEM are still there. So it had just been installed, and probably we were the second or the third ones to use it. But we actually waited for it to be installed for more than 2 years. Okay, let it be installed and then we will use it.
Let somebody use it first.
Yeah, yeah.
That's good. So, like, since you mentioned about your guide and your lab, it was quite good. So, like, what do you see when you guide your PhD students? Like, do you have his his shadow sometimes or something like that?
Yeah, yeah. So sometimes my students call me mommy, because like flow mein. So yeah, I do carry my Professor Al Kothari's shadow, and I see to it that my lab is also a family. And I'm trying at least my level best. Although now that I've become the coordinator of the biotech program, I don't give enough time to them. I wish I could read more for them. But yeah, yeah, apologies to all my students. Because there is a lot of departmental work that I have to do.
Uh,
So
yeah, I see that. But I'm sure your students might be quite happy in the lab.
yeah, I think so.
Of course. So, do you have mentors in your life? Like back then, apart from your PhD guide and your maybe professors, did you ever had mentors? Like uh, maybe it could be your father as well because he gave you a very good advice. So, like, did you had different mentors in part of your different life, or like how do you see it now as well?
Yeah, so as you said, my father has been my constant all over, and he's still there. So any big decision in life, he has to be asked first. So about buying a house or buying a car, anything, it has to run through him. And in fact, my husband also has his father, so we have two fathers now. So both the fathers are like our mentors. But yeah, regarding science, our professors have been really nice. So even after I joined Neela Devi, I remember coming to Professor Anjana Desai, Professor G Archana, because we had certain projects in the hospital where uh, I thought their inputs would be helpful. So yeah, time and again, I think we've asked, even today, like if because I am the member of the board of management, and I have a lot of other responsibilities now in the university. I do ask. I have to ask, actually. With I don't think I can survive without their help. Professor Dr. Pranav Vyas, he helps me out. He gives tips as to how to go ahead. And Professor Anjana Desai, Archana ma'am, all of them are there. Anuradha Nerurkar. So they're all there for me, and they give. They're just a phone call away. So they give me tips as to, because they are all seniors, so they have a lot of experience, and experience has a great value. So
Absolutely. I still remember when I was graduating, I was about to finish my master's. I had a talk with Professor Pranav Vyas for 1 hour,
oh really?
and that somehow changed my perspective with towards science. But I really like talking to him. I wish
Right.
I could catch him again when I visit.
Yeah, he's superannuated, so he doesn't come so often to the department now.
Yeah. I had to catch him. Like I waited for 1 week, always going here and
Yeah,
there.
he doesn't have a phone also, so how do you catch
And
him? He
I
still
think
doesn't have a.
exactly. I used to go to the underground lab. I think not underground, but on the
CFL.
ground floor. I
CFL.
think.
It's called the ground floor
Ah,
lab.
no. So I used to always go and maybe wait for some time, for half an hour, 1 hour. He was not coming, and then I was like, okay, अगर वो आए तो call करना मुझे.
Right, right.
But yeah. Um, and okay, so I would like to ask you a question that what is the biggest highlight of your research career till now? Like, uh, I would say, yeah, or the best achievement. It could be getting a PhD, of course. That's one of the biggest achievement. But apart from that, like, I would say, let's say maybe some of your research article or which is like memorable that this one was game changer.
Uh, I think it's yet to come. So we do a lot of genomics, and we are hoping that we will use this information. And recently we've started using CRISPR in the lab, and we've also started using microfluidics in the lab. So I think it is yet to come. We have not even published more than 5 impact factors. So I don't think the best moment has come for me. I aim for a JBC one day, Journal of Biological Chemistry. So when I'll publish in
Mhm.
JBC, I think that will be my best moment, according to me.
So you still get what dream.
Yeah.
That's really good. That's really good. Okay. So, like being a PI, what advice would you like to give to, especially master students who are doing their masters right now, who would maybe start their research journey or already have started in their thesis? So, what is like the three advice that you will always give? I'm counting on three because it would be easier for you to think.
So, what I see, at least in India, because most of us are Indians, they all have joined over here, we tend to do repetitive work. We tend to copy somebody's work and try to do the same thing, hoping that we will get some new result. So, doing the same experiment that others have done is not bad. You will learn a skill doing that, right? But then you should be able to comprehend the existing literature and find your way out of it. You might get into a trap of the repetitive result and doing the same thing over and over again and not find a way out. So if you want to find a way out, and you have to ask the right question, and you have to be able to find out something new of your results and make sense of your results. So one, obviously, the lab skills. If your hands are good, and if your data is clean, you have a long way to go, I'm very sure. You are ethical and you are not, tempering your data, then again, you have a very long way to go. And third, you have to sit down and write. So you cannot put the data in the drawers and do nothing. You have to compile that data and start writing, which is what we don't do. There is so much of data and people just sit on it and don't do anything about it. So unless you write it, you will not know where you stand. So I'll say that just even if your manuscript is half, start writing it. You will find another direction till you finish the manuscript. So yes, for the young generation, lab skills are a must. You should have good lab skills. You should have good calculation skills. I know there are many tools nowadays, but still you should know the basics. And you should have a good communication writing skill. All these three skills are very important.
I think the first advice really hit me since I'm a first year PhD student. I'm taking some papers as base, trying to see if data I can generate that and I can move ahead. And I'm struggling in that loop,
Right. I
trying to
know how
find
it.
out something out just out of, let's say they have published in molecular cell and I'm trying to replicate that, but somehow there are some technicalities which I cannot understand and I'm now trapped in that loop. So first I have to generate the data that they have and then I can start ahead. Or maybe generate the data that already lab has from previous people. So it has been a non-ending loop. Yes, absolutely. Okay. So since you mentioned that you started with microfluidics and CRISPR, I mean CRISPR nowadays everybody is using microfluidics. People have started to use a little bit, and nowadays people are also using this lab on chip method, you know, where they have organoids, but these organoids are basically on the chip and they are connected with the microfluidic system. So how do you take up these challenges? Like, since this is also very new for you, right? And like a scientist, how do you see it right now? And also, how do you see your students would be learning those things?
So in my lab, there are certain. So my lab works on 4 organisms, and I'll share the presentation when the time comes. But I have this simple theory that every student has to standardize a new technique in the lab. So if somebody is doing whole genomics, then that student has to set the pipeline of whole genomics and teach the rest the pipeline of genomics. If somebody is doing CRISPR base editing, then that student has to completely validate the whole process so that the rest of the lab can learn. Likewise, if somebody is doing microfluidics, so any new technique, it will take time to set up. At least like in our experience, CRISPR, this time it's the third dissertation student with one PhD student, and now we've started seeing the results that okay, now it is working. And so it's not easy. Microfluidics, the pumps and everything is so costly. So it's not easy, but you have to take up the challenge. No, if you keep cribbing that, oh, ye to nahi hai apne paas. And so facilities, I never see facilities as a limiting. You cannot complain that I don't have this instrument and that's why I'm not doing this thing. That's a very lame excuse to make. You have to make efforts to get it done. Or e- e- I mean, I don't know. That is always that has always been my logic. So in my lab, right now there are 4 of them full time. uh, all the 4 topics are different, and you will see they are different. So it's not that the same everyone is doing the same thing. That does not happen in my lab.
I see. Absolutely. I think this is a good strategy that a student will set up a complete new technique. Okay. So, talking in the same direction, how do you envision your research or your lab in next 10 years?
Right.
Publishing a
So now,
JBC?
yeah, that is of course the main goal, publishing a JBC, because I have now realized that just publishing is not going to help. So publishing is fine, but we would also like to make a kit or something for AMR. So we are working in that direction. So we would obviously want to patent something and device kit or a device lateral flow assay for diagnosis of AMR. That is the ultimate goal, actually.
Then we would have Professor Devarshi Gajjar on Shark Tank India.
Yeah, maybe some of my student. I don't know what I. Yeah, let's see.
Maybe you can join them like a mentor.
Yeah, yeah, certainly.
So, like, now I'll ask you, since I have started asking you few questions being a PI, uh, I would ask you one more question on this. Like, what motivates you to pursue research ahead being a PI? You know, you have a lot of responsibilities right now on your head. You have your family, you also have your so many students to take care since you are coordinator of biotechnology program. You also have other things going on, and then the research. So what motivates you to still, go to the lab and read the papers, even at this point having so much load?
No, I think to be a good teacher also and to remain, I don't know whether I'm a good teacher or not, but to teach decently, I have to remain updated, and to remain updated, I have to do research. That is the so. Let me tell you, all of this experience that I had about the virus purification or about the SNP analysis that I did at Ila Devi. We did so much of SSCP, PCR, and I used to do so much of PCR, so much of gel running, and doing molecular markers in patients. All of that has helped me in teaching. So if I had not done some decent research, I don't think I would have been able to teach the way I'm teaching today. So even to teach in a decent manner, one has to remain updated, and to remain updated, doing a little set of research. So it's learning by doing. You cannot just read and you feel that okay, now I know everything. Unless you do it, how will you be convinced? So I am totally off, because our skill, life science, biotechnology, cell biology, molecular biology, is all a lab practical based skill. You cannot just learn it by reading. So you have to do it. You have to do the experiment. So yeah,
That's true.
I think I have to balance it out. If I want to teach well, I have to do research. Only then I'll be able to, uh, balance both the duties.
Just a very quick question. I just I just remembered something, and fine. Uh, when was the last time you holded a pipette in your hand?
So I take MSc practicals, right? So
Ah, so you
I have to put slides on the microscope. I the initial so
have
when
really
the new
good.
batch comes in the biotech, I am the one to interact. So I have to teach them ripening Petri plates. I have to teach them ripening the glass pipettes, putting things in the autoclave. So I do staining. So I mount the fungi. I put lactophenol. So I think I have you. I do.
Very
It's not that I've totally
unlike
forgotten about it.
that. That's really good because there are, I would say, many PIs who are just out of lab contact completely. So
No, that's not possible in MS University. I don't think that is ever going to happen in MS University.
that's good. That's really good. So I would like to ask you a second last question. We would give a last question at the end. Uh, the question is that being a female PI, as you mentioned earlier as well, even when the placement things come, what challenges do you see being a female and pursuing career in STEM? And yeah, first question I would ask is what are the challenges and also drawbacks?
So one of the very first challenge that I faced, and I'm sure every mother would face. See, doing the household chores is not a very big thing. You can hire somebody and get things done, and staying with in-laws all that is just a passing of time, and you can adjust, and everything is possible. And nowadays, all the people are also quite adjustable, and they allow, like, not allow, but they in fact contribute in the uplifting of the bahu or whatever. So that has never been a problem with me. We have had maids always in our houses. All my co-sister-in-laws are also working, so that has never been a problem. The biggest challenge in a woman that comes is when she becomes a mother. So that guilt that we take on, and that guilt goes on till your son goes to college, because throughout, men unfortunately don't feel as much as we do, obviously, because our children are a part of our body, they've come out of us. So that guilt, that dealing with that guilt, and even when I used to go for a conference, I remember my parents coming along with me with my son, just so that they can take care of him. Every year we used to go to this Indian eye research group at Hyderabad. I'm still a member. I still go every year to Madurai and to Hyderabad. But they used to come specifically just to babysit my son. So that guilt is there, that you always feel that you have not given your 100% to your child. But as the time passes, your children, or they will value your sacrifice, and they themselves will tell that, no mama, whatever you have done, you've done for me. And they appreciate your career also. They might not appreciate when they are young, or when they are too small in their teens and all, but the moment they cross out of college, or they are in college, they themselves come forward and say that, no, oh, you are doing so much. So just wait, all the ladies, just wait for the right time. And even if you are guilty about getting married, or about having a child and not giving your 100% to your partner or to your child, you have to deal with it. And it's fine to have that guilt, and you just have to overcome that guilt. That is all. Otherwise, I think now people are changing, and people are accepting a lot of changes. I've seen so many in my society. The men do equal work as the lady does, because more and more couples are both are working nowadays. So and the people are not judging them also. So I think time has to change, and you guys have to change. There, you all, your youngsters, those who are listening to me, please come up with a change. You have to change, and only then our country will change, and the ladies will be happily going to work without stress, and women will happily go to work without stress. Yeah, I hope I answered your
Absolutely.
question.
Yes, absolutely. I would say that I love cooking. So, and I mean, even if I don't love, I have to cook for myself here in abroad. So nobody will come and nobody will clean my utensils.
So the problem is not for people who are living out of India, the area. The problem is for the
Yeah,
people who are living in India. All those who are outside, that's a different world altogether. All those who are in India, that is a different world. And the same people, when they come to India, they will not do anything. So that is a very tricky scenario, but it's there. I mean, it is there.
yes, absolutely. I think
Our society is biased towards patriarchy, and so
absolutely. I I would definitely agree on this somehow because there comes a point in every woman's life, as you mentioned, that they have to choose. Sometimes they are given an option to choose.
yeah.
So.
No, but
Actually,
if you talk it out, you can solve the problem. You have to talk and you
that's
have to
true.
convince the people that my career is as important as my family. So if I pursue my career happily, I will even do all my duties for the family. Like, it's not at the cost of something, So the family is not at the cost of the career, and career is not at the cost of family. You have to do both. You have to balance.
Absolutely. 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 Okay, ma'am. So we finish with such short question answer round. I think it was long, not short,
Yeah,
but uh, thanks for all your answers. I think youngsters really got, even me too, including me, got good tips from you. And I think now we would like to hear your presentation and about
yeah,
your research that goes on in your lab.
sure. About 10 minutes. Is that fine? Till
Yeah,
11:30?
yeah, yeah, absolutely. It's fine. And then we would have a short audience question for 5 minutes, and then
Sure.
we could end.
So I'm sharing my entire screen, right?
Yes,
Is it seen?
yes,
Okay, I'm audible, right?
yes, ma'am.
Okay, right. So first of all, thank you very much, Dhairya, particularly Dhairya, to have me here, and it is really a pleasure to interact with the youngsters and to share my story. Because it is important that all of you must get some tips about how to go ahead, and I'm always there. You can reach me out at my email also. So this is my lab. It's Microbial Pathogenesis Lab at MS University of Baroda at the Department of Microbiology and Biotechnology Center. And I've been here since 2013, so it's 10 years now. Uh, and we work on four pathogens, as I said. We work on the antimicrobial resistance and virulence of Fusarium species, particularly the species that cause corneal infections. Then we work on polymicrobial biofilms as well. So we work on catheters and the organisms that are housed on the catheters. And we work on these two bacteria, which is Klebsiella pneumoniae and Pseudomonas or aeruginosa. And when I share my work about each one of them, I'll also tell you why each one of them is very important for us. So let me start with the first one, Fusarium. So we particularly right now are doing genomic and transcriptomic analysis of the Fusarium, but let me tell you why and how did I get into this. So when I was working at Ila Devi, we used to get a lot of corneal samples, keratitis. So this is the clinical picture of keratitis where the person's cornea gets infected with the fungus. It becomes opaque completely. There is a lot of inflammation. It is a very fulminating infection, and the infection could last for more than a year. And if even if it gets cured, even if the fungus gets cured, then the cornea has to be transplanted as shown over here. Now, how do these patients get the fungus on their cornea? So let me tell you, India is a tropical country. We are an agrarian country. So when the farmers are harvesting their crop, unfortunately, they don't wear any protecting glasses, and the spores from the fields, the fungal spores from the fields, by chance. Get attached to the cornea. If the cornea is abraded or if the cornea has a scar, then the fungus is able to cause infection. So it is a rare infection, but because it is very serious, we thought we should do. And when I was in Ilya Devi, we actually collected a lot of samples from patients. And I came to MSU and I bought all those fungal samples to MSU with an aim that we will try to characterize them. So initially we just did some microscopy of how the spores look like. Then we also did some antifungal susceptibility as to find out how resistant these fungal cultures are. And while we were doing that, we realized that okay, just finding the resistance is only, and that time 10 years ago the whole genome sequencing was picking up. of many, all the model organisms, what it was over, and it was like kind of becoming cheaper and cheaper. So therefore we thought, let us go with whole genome sequencing to find out what are the different ways by which fusarium becomes resistant. So it could be target overexpression, it could be target incompatibility, stress response, drug changes in the drug transporter, and or cell permeability. So any of these could be primary resistance, or the fungus can acquire can acquire resistance also. And all of this is easily possible when we do whole genome sequencing. And by sequencing, we could able to characterize one such mutant version or mutation in the CYP51A, which is responsible for the cell wall synthesis. It's an enzyme. So we could find that the drug is not able to bind to the heme, and that's how the fusarium has become resistant. This, of course, is unpublished data. We'll be publishing it soon because we are about to publish and compile the genome data also. So now that we got this, we are trying to do drug repurposing also, that what are the existing drugs that are approved that could bind to this particular mutant version, and can we use this drug instead of the existing antifungal drugs. So alongside genomic analysis of fusarium, we thought that there is no model of keratitis. So there are animal models, but there is no actual in vitro model of keratitis. So then we thought, let us just try making a caprine cornea model. Caprine cornea means the goat cornea. So what we do is we get the eyeballs from the goat, from the mutton chop. We dissect the cornea and we culture the cornea. And in this first paper of ours in 2018, we first proved that yes, the cornea survives, it's healthy, it's transparent for 15 days. This is the MTT assay, and we established the model. Uh, uh, we established that the explant culture of cornea is possible. Then what we did was that we infected the fusarium spores, like you can see over here. This is day 2, day 4, day 8, and you can see these are the fungal hyphae as shown over here, that the fungi is penetrating. And so this model is published, and we are now trying to characterize these virulence factors using transcriptomic analysis of this model. So this is about fusarium. And more importantly, why fusarium is important is because it is more prevalent in temperate countries, tropical countries like India, and not much research is being done in the West, because it's not a very big problem over there. It is a very huge problem in India, China, and all these countries, because we are agricultural related countries. Okay, so next I would briefly touch upon polymicrobial biofilms. So here what we do is we get catheters from hospitals. We try to culture how many bacteria are there. We try to identify what are the different kinds of bacteria, fungi that are housed on the biofilm. So you can see this is the biofilm on the catheter, and how does what is the different proportion. So what we found was that in more than 50% of the catheters, we always found that the candida was always associated with either Klebsiella or with E. coli. And so the candida was never alone. The bacteria were alone in some of the catheters, but the candida was always along with a bacterium. And so that's why we started using microscopy. We make the biofilms in these flow slides, and we try to do live dead staining. So this is the picture of calcoflow showing the overall morphology of the biofilm. This is the live dead staining of Candida tropicalis plus Klebsiella pneumoniae. Likewise, this is the live dead staining of Candida albicans plus Klebsiella pneumoniae. And again, this is the live dead staining of Candida tropicalis plus E. coli. So when you compare this and this, you can see that in presence of tropicalis, the E. coli is dying, because you see more of the red color. That means more of the PI staining is there, but the green color is less. That means the live cells have reduced. So likewise, we did this in the static And now this is our microfluidic setup. We have connected all these pumps and channels, and we are going to do the similar study using flow in flow conditions. And this is the IBD slide that we use for mimicking the urine flow, because we work on urinary, all these cultures are causing urinary tract infections, and therefore we would like to see their behavior, that when you treat them, how are they released and whether they can migrate up to the bladder, etc., etc. So this is what we are doing about polymicrobial biofilms. Now about Klebsiella pneumoniae. So why Klebsiella pneumoniae has suddenly come into limelight? It has also been reported in the critical priority pathogens of WHO. So before 15 years, not much was reported about Klebsiella pneumoniae. When we started in 2016, there were very few papers in PubMed, and we thought that, okay, let us see what happens. And in the next 5 years, there was so much of published data on the microbial resistance profile of Klebsiella pneumoniae. So what is interesting about Klebsiella pneumoniae is that it it takes mobile genetic elements from its neighbors, and it can give away these elements to its neighbors also. So basically it's the root cause of the spread of all these antimicrobial resistance genes, and that's why like people are studying it. We are also trying to study what is the scenario in India. Now, what we do is we obviously collect different samples, we do the resistance profiling. Of course, we want to make a diagnostic kit, but we also do some phagocytosis assays, some biofilm assays. But right now, I'm just going to share about the carbapenem resistance. So we are particularly looking at carbapenem resistance. And now why carbapenems is, so beta lactams, as you know, are cell wall inhibiting antibiotics, and they are the largest class of antibiotics and the most frequently prescribed class of antibiotics. So compared to the tetracyclines, fluoroquinolones, and other antibiotics, beta lactams are the maximally prescribed. And because they are maximally prescribed, obviously the resistance to them is also high. So, out of the penicillins, cephalosporins, carbapenems, monobactams, why carbapenem resistance is important is because when an organism is resistant to carbapenems, we are literally not left with any other drug to treat the patients except colistin and tigecycline. So, therefore, finding out whether the organism has carbapenemase or has the gene that is responsible for carbapenem resistance is extremely necessary. So what causes carbapenem resistance? So you can see there are many enzymes that will cleave penicillin. So this is a chart of beta lactamases. You can see there is class A, B, C, and D. And the red ones over here are all carbapenemases, which means you can see there are so many enzymes that could cleave the carbapenemases, and all these enzymes can cleave almost all the earlier drugs. So if you summarize this, if there is a penicillinase, it will just cleave penicillin. If it is a broad spectrum beta lactamase, it will cleave the first generation cephalosporin. AMC will overexpress and cleave all of these. Likewise, you can see the class B, class A, and class D carbapenemases will be able to cleave all these antibiotics, and practically the organism is resistant to all of these, and it can be called as XDR, extremely drug resistant, or time drug resistant if it has resistance to colistin and tigecycline also. So we are interested in the genes that are responsible for carbapenem resistance. So all of these KPC, WIM, IM, NDM, which is New Delhi metallobeta-lactamase, OXA, which is oxacelinase, all of these are genes which cause resistance to carbapenems. So what we do whole genome sequencing, and then we find how many genes are there, whether they are on a plasmid, they are on a chromosome, how many prophages are there, how many IS elements are there, transposons are there, how many integrative conjugative elements are there, etc., etc. So for example, we published one pan drug resistant organism, and we were able to find that this organism has 5 different plasmids, and it has multiple copies of NDM, multiple copies of CTXM, and that is how it has become a pan drug resistant organism. So you can check these papers which we have published recently, and all this information is there in our papers. So the last one, Pseudomonas. So how Pseudomonas is different from Klebsiella? So Klebsiella is a source of drug resistance. Pseudomonas is highly virulent. So if you see the resistance, if you see the virulence factors of Pseudomonas, there are so many. You name it and it has. So Pseudomonas is it is a highly virulent organism, and therefore any infection with Pseudomonas, it will progress very fast. In no time, the whole organism or the whole body will be spread of Pseudomonas. And so Pseudomonas is important with respect to its virulence factors, whereas Klebsiella is important with respect to its resistance profile. So what we do, again, we try to collect isolates and we try to characterize the virulence factors. One of them is the biofilm formation. So this is the scanning electron microscopic picture of a weak biofilm forming Pseudomonas and a strong biofilm forming Pseudomonas. So you can see the strong biofilm forming Pseudomonas releases a lot of extracellular matrix, EPS, exopolymeric substances. Whereas the weak biofilm, you can see the Pseudomonas rods shown over here. So therefore, that's the difference between the weak and the strong biofilm. Apart from that, we also did some adhesion assays. So you can see the strong Pseudomonas is adhering more compared to the weak Pseudomonas. And these are some of the videos. I don't know how to switch on the video. Yeah. Yeah, you can see it's on. So you can see the twitching motility over here. The cells are moving. So we proved, actually, so if you see very carefully, the cells are moving over here. And it is the twitching motility which is causing. So this is of a strong biofilm, and the next one is of the weak biofilm, where you can see very less motility in this particular slide. So we published a lot in Pseudomonas also. Like, you can check these papers on biofilm, persister cells, and Pseudomonas. And so in this paper, we showed that cell adhesion and twitching motility are the main factors causing strong biofilms in Pseudomonas. Right. So I would be happy to take up some questions. This is my lab currently. Siddhi and Swati have already graduated. Hiral has submitted and will be defending soon. This is Suraj, Milan. So the most of the cornea work I showed was of Penal. The catheters and the biofilm work of microfluidics was of Purvi. Klebsiella was together Siddhi and Haley and Suraj. And Hiral and Milan are doing the Pseudomonas work. Tosin was a TWAS fellow who visited our lab last year. These are some of my collaborators, Pushpendra, Ratika, and Sonal. And these are some of my funding that I've received: MBO, GSBTM, DBT teaching program. And not to forget all my clinical collaborators, because all the samples that we get are from these collaborators, and which is they help us a lot in understanding the clinical problem. So, lastly, I'd like to say this quote, that a mind once stretched by a new idea never regains its original dimensions. So, I strongly believe in education, and you cannot undo knowledge. Your grades will be forgotten, but the person you become during the learning process lasts forever. So, at MS University of Baroda, our motto is to imbibe values of hard work, discipline, and team spirit in the students, to make them able to survive and thrive, to compete with full ethics and values, and to make them independent and then interdependent. So thank you very much, Dhairya. This is our beautiful dome, and if somebody wants to come to Baroda, please come and visit us. I'll be happy to take questions.
Thank you, ma'am. It was a really, really good talk, I would say. I am not much into microbiology, but I could understand of it.
Yeah, I would like
I
to ask one question.
Yes.
And Satya,
Yeah, hi there. That was a very wonderful talk, ma'am. So
Can you just
I would like,
introduce yourself? Can you just
yeah,
tell me?
sure,
Yeah.
sure. So I am Satyarth from Delhi. I am working as a
Okay.
project associate in CSIR Institute of Genomics and integrative biology, and my
Okay.
master's was in microbiology, and I'm part
Okay.
of the SciComm team.
Great.
So I would like to ask that we see AMR is going to be a hidden pandemic in the coming future, right? And
Yes.
people are still trying to, I mean, get some therapy like bacteriophage therapy,
Yes.
etc. So how do you see AMR in future, specifically in India? Because there are no regulation bodies who can control the uh, antibiotics and all. So how do you see AMR in future? And are there any ways to prevent it? Are there any effective strategies by which we can prevent it as a researcher?
Yeah, so one of our motto is to educate people about AMR. So uh, the basics of taking drugs, not to leave the prescriptions halfway, not to pop pills unnecessarily. You can't take azithromycin just for a small sore throat. So all these are, we do it at our MSc level to all our students. All my batches get this one lecture from me. But yeah, at India level also, there is a national program of AMR. All state-wise programs of AMR are going on. Like in Gujarat, I am also a part of the network program of AMR. Karnataka also has come up with one. Kerala has come up with one. They are monitoring the bugs very closely. A lot of genomics is going on. And I can't say if we can prevent it, but I can at least say that we are ready to handle it. And phage therapy has also approved, like you must have heard of the Johri fellow, Johri, who from Vitila's therapy. So yeah, I think it is a difficult task for us, but it is not something that we cannot handle. And if we have enough awareness, then of course we will be able to tackle it with great care, I think. But yeah, there are too many genes floating around, so I do understand that it is a critical situation.
Thank you. Thank you.
Thank you, ma'am. So I would like to ask one question. The one last question that we were hanging on. What is the uh, what is one of the best discovery of science that you think?
Oh yeah. So according to me, PCR, I cannot live without PCR. I cannot imagine my life without PCR. So I think that has revolutionized the field of doing science. So of course all the others are equally good,
Absolutely,
but
absolutely.
One
So
student,
we had
yeah, one student has asked a question that how to can you suggest how to write a review article? Which point is necessary to write a review article? If you can answer, ma'am.
yeah, so first of all, if you want to write a review article, you have to jot down a few keywords and do a quick review of literature to find how many existing reviews are there with those keywords. If you find there are already too many reviews with those keywords, then I don't see a point writing a review. So you first have to be convinced that there is no review in the area that you are thinking, and then start writing it. And then collect all the information, bifurcate all the information into different parts, and then I think making a splitting the topic into various topics will help in organizing the entire review, or doing a mind map kind of a thing. Remember in school they used to teach us. I don't know if I answered it correctly.
Yes, ma'am. And
yeah,
one student has asked how much GPA matters.
Anything above 6 is fine, according to me.
Okay.
In MSU, yes. Okay. So I think there was one very interesting question that somebody asked from UG category.
Mhm.
The question was actually framed a bit differently, but I would like to ask that. So the person asked that who controls our body, microbiome or our cells? I couldn't understand this question, but I think the person is trying to connect it with neuronal regulations of microbiome. That's what I could think.
Yeah, right now
So
of microbiota is being linked to everything actually. The bio, but I am not very well aware of that research topic. So, but there are a lot of papers and reviews coming up that the microbiota is sending some kind of signals to control all the behaviors. So, yeah, I think it is a very upcoming field, and a lot of work has been done. So, somebody is asking, ma'am, how to isolate viruses from soil? We cannot grow them on Petri plate on agar since they are not active. So, which kind of a virus do you want to isolate? You want to isolate a phage for bacteria, or you want to isolate a virus which is infecting a plant or infecting an animal? It depends on what you want to isolate. So, if you want to isolate a virus infecting a bacteria, it's very easy. We do it in our class practicals. I'm not sure soil would have so many viruses, but effluents and the dirty areas will have a lot of viruses. So we usually do phage isolation from these samples, from the effluents and the nala and all those places. So there's this Nidhi Mishra. I'm a student of first year, and as we are at a stage of looking for internships, and ma'am being a PI at MSU, I wanted to ask, what about one should go through with? What I wanted to ask about what one should be thorough with or be prepared with and know about already before applying at labs. I see about internships. If you write to 100, you will listen from 10. So please be prepared to write to 100 people and hope that at least 10 of them will reply. That's the strategy we use at MS University of Baroda, because all our biotech students also need internships, and it has become very difficult because all of you are doing internships. So how many people do you think can do internships? But because of these times, all of you want to do internship.
yeah, it's very competitive.
So Krish is
Uh,
asking, hey everyone, my question being a pharma student was that how frequently can we apply such microbiological studies in our research to influence our drug formulation? Could you please note down a few general references for us to look into? I suggest, Krish, you please find the correct collaborator who will do things for you, because as a pharmacy student, the learning curve will be a bit longer. So you might have to end up spending 6 months in a microbiology lab learning all the pure culture techniques. So rather than doing that, find a person who will do it for you, and that will be easy. Varun Varadarajan is asking, hello, ma'am, can you please suggest how to explore or go in depth on a particular topic, say pathogen of soil? I would suggest please write short questions so that it's not very difficult for me to grasp.
I think I will just quickly say that. So the person is asking that uh, how can how to go into depth of particular topic, like a general topic,
How to go into the
from where to start.
so I don't know if from which college are you in BSc or MSc, but if you can start with reading a latest Nature Cell or any paper, and then you have to follow that topic. So what I generally suggest my MSc students is that, let's say in semester 1 or 2 you have read a research paper or you have presented a research paper as a part of our curriculum, then you choose a topic which you will follow it up, so you can keep reading about that particular topic throughout your MSc, even after your MSc, so that you build that knowledge with you. So, on the first place, you have to read the right paper and the paper which you like. If you present something that you don't like, then it'll be like you will just prepare some slides and present it, and you will be marked for it. So, I always ask students to follow your liking of topic. Just follow it up. That means uh, keep reading and keep following that person, that researcher. Okay, okay, he has published this today. Next, what is he publishing in context to what he has published today? So, Jiljil is asking, ma'am, what is your turn on mathematical biology in future of biomedical science? Of course, it is very important, but you need to find the right collaborator. I always feel that I
Yeah,
cannot do everything myself. So, it is not right that I also don't do and I don't allow others also to not to do. So, it's always good that whatever I feel I cannot do, I can ask somebody else to do it for me. So yes, of course,
yeah.
AI is coming in a big way for us. I think I truly believe that AI
Um,
is going to make presentations for me in the near future. I have not used it so far. People are using it, but yeah, I feel it is coming in a big way for us too. So Devangi is asking, how much role does your dissertation play in context with research, as in the MSc dissertation?
I think so.
Yeah, so your MSc dissertation, you have to work, you not work, you have to slog. You have to slog like anything. And if you get habituated to do the same experiment 100 times, that means you are the right person to do research in future. If you get tired in 2 times, and if you get tired, a result nahi aa raha hai, then forget about it. You're not made for doing. You find a job in a pharma industry and do QC QA all your life. So your MSc dissertation is a testing ground. I always tell my students it's a testing ground of your perseverance, of your patience. And if you can, like I remember, I used to do protoplast fusion and I used to make 100 Petri plates in one go. Just sit down, one full round of autoclaving, and you pour one after the other. And I used to like doing that, making 100 Petri plates. So you have to first keep that in mind, that you doing it as a burden, then don't do it. Then you are not you're not suited for it. So your dissertation is a like pilot scale PhD. I always say this. It's your testing ground about your skills, your hands, your brain, your ability to ask questions, your ability to defend. Everything is your dissertation. So please do it very religiously. Don't feel कि अरे 2 स्लाइड बन गई है बस अभी कुछ करने की जरूरत नहीं है. That's not the right way to do dissertation. Some people do it. They feel कि 2 स्लाइड बन गई है बस अभी पास तो हो ही जाएंगे. That's not very nice. That's not good. You will face the consequence in future. And if
I think that you, I can relate.
yeah,
I had, I remember my thesis days.
Jill is asking why India is lagging behind other countries in research. Well,
Hmm. I don't think India is lagging behind. I would say India is doing much great considering the socio-economic statuses, the problems
so
that we have in our country.
right. So we have switched gears. Like in India, we have been asked, and people feel that we should identify problems related to India and address our own issues rather than solving issues of the Western world. So the research grants that are now given to the Indian scientists is a little bit diverted from the earlier time, that they insist that okay, how is it solving one of the major Indian problems? And therefore, I will not say that we are lagging behind, but because we are switching gears, it'll take some time to come to the log phase and speed up, go into the next speed. I think we are already in the speeding mode. So yeah, the next 10 years will say how good or bad we are. So one, of course, is the money. We have to invest a little more money. The country has to invest, but yeah, we are still doing good from whatever we have, and the best is provided to us in the existing scenario. So Sagar is asking, I'm working in Amul R&D Federation as a research scientist. Is it possible to pursue PhD? Of course, it is possible. Come to MS University of Baroda. We have an executive PhD. If you have 5 years of experience, you can register with any of the professors. You can write to me also if you want. If you don't have 5 years of experience, then it is not an executive PhD, it is a regular PhD. But in that case, you will have to figure out how will you work and where you will work. In an executive PhD, the understanding is that you are working in your working place. So Amul is giving will give us a no objection certificate, that Amul has no objection in you doing a PhD with someone. So it is certainly possible. Many people from Sun Pharma also have been doing PhD with MS University. So Sagar, please write to me, and I'll be happy to help to you. So Ankit is asking, what things do you think most PI looking students for hiring a GRF? Yeah, so it's some basic understanding and some lab skills. More importantly, basic understanding is more required. You should know the fundamentals of biology, and if you know the fundamentals of biology, then yes, you will be far better. The lab skills can be taught. It's not that one cannot learn, and it just takes 6 months to learn a particular, let's say, cell culture. Nowadays most of the students feel, अरे cell culture नहीं आता है तो कुछ भी नहीं आता है. ऐसा कुछ नहीं होता. Cell culture is just a technique. You will learn cell culture when you join a lab, but the fundamentals of cell culture, you read Freshney, you read textbooks, that will take you farther than just knowing a technique. So Sandeep is saying, very nice informative session. Thank you very much, Sandeep. Krutika, thank you so much. I learned a lot today. Practical, it's really nice. Thank you very much, Krutika. Nimish is saying, what are your suggestions on doing masters? Like, it is good to do it from India or from abroad? Oh, that's a tricky question. There you can ask. I live in India. I have never lived abroad, so I cannot answer that question.
That I would say that depends upon your priorities, that what you want to do. But I would recommend to do finish a master's in India definitely, because I did that. MS University is providing quite good education. And ma'am, since you were telling if you are a good teacher, yes, you were a very good teacher. We really enjoyed your classes.
So,
So
Nimish, money is very important, right? So, if you have enough money and if you have sponsors, go abroad. But if you, I mean, that's a very tricky question. Even all my students ask, but I leave it to your fate. And I don't know there is an ideal place to do masters. I don't think I'll be able to answer that. Okay, Bhumika, you are a great motivation. Thank you very much, Bhumika. Shweta, thank you for the wonderful talk. Kiran, thank you for such a wonderful. Can phage therapy be used? Yes, certainly it is being used. But the only thing is that we don't manufacture the phage over here. It comes from Belgium. Right now, the company is in Belgium, and Vitelas therapy gets the phage from Belgium. And so, is there a side effect? Yes, there is a septic shock sometimes, but then all that is monitored, and now all that is standardized. So they give the dose in such a way that you don't reach that shock kind of a stage. Ankit Yadav, for sharing research. Ankit is our own Ankit. Thank you, Ankit, for joining.
okay, so I think I will have to interrupt in between, ma'am. Very sorry.
Yeah.
I think we are already out of time. We would have
Yes.
ma'am again,
Yeah.
definitely, and we are also gonna have your collaborators very soon.
Oh, really? Yeah, you can have
Yes,
Pushpendra and Ratika.
I have. I've already
They're
emailed them.
yeah, great.
Already emailed them. I'm looking
You sh-
forward. So
you
it would be
share
really,
this thing to me. You share their.
yes, I will definitely.
I'll attend theirs. Yeah, I would like to attend Pushpendra and Ratika's. They will
Yes,
be very motivating. Let me tell you, they are
yes,
very good researchers, and so see, the moral of the story is that biology is not a very lucrative stream as engineering is. So if you are here for money, money will come, but it'll take some time. Just have some patience. Initially, you just have to toil your way out. It's not like computer science graduates of IIT that you will get packages of 15 lakhs and 20 lakhs. You will hardly get 3 lakhs or 4 lakhs after your MSc. right? But once you have some experience, then of course the future is very bright. So just trust the process. Just trust the process. I always say that to my students.
I think that could be the concluding remarks of
Yeah,
the entire session today. Thanks for this wonderful talk, ma'am. We would be very happy to have you future as well. Again, maybe on some other topics. And would be good if you also associate with us and also help us to invite more speakers upon. So, since many people were asking about the other events that we would have, yes, we would be having such motivating events ahead. Uh, we would request for that please fill out the feedback form. We have sent it in the chat box. It would be important for you so that you would receive the necessary information, and we would also have your email addresses and everything. So please fill out the this form, feedback form. We will also get feedback what we want to have to improve. We are just starting, so we don't know. Maybe, ma'am, you can also say what we
sure,
need to
sure.
improve.
Certainly, I'll get in touch with you. Thank you. Thank you for such a
Absolutely.
large turnout. Huh? I thought 10, 15, 20 log honge. I never knew
Mm,
so many people will come.
we had more than 200 registrations.
Really nice. Thank you all. Thank you. Thank you.
Thank you so much, ma'am.
Yeah, please come to Baroda and visit us. It is a nice
I
place.
will. I am planning in October. I will
Sure then.
definitely.
Sure. See you then. Yeah. Okay.
Yep.
Thank you
Thank
all.
you,
Have
ma'am.
a good Sunday. Thank you very much,
Yes.
Dhairya. Thank you.
Yes.
Shall I
Thank you,
leave?
everyone, for joining. Yes.
Yeah. Yeah. Thank you.
Thank you.